Imagine you're stepping into a bustling laboratory where scientists, driven by curiosity and a desire to save lives, are peering into microscopes and meticulously testing samples. It's the early 1980s, and researchers are zeroing in on a persistent viral threat known as Human Papillomavirus, or HPV. They discover that it doesn't just cause warts, as many had assumed. It also has the insidious ability to trigger certain types of cancer, most notoriously, cervical cancer. In particular, two strains of HPV, types 16 and 18, stand out, causing about 70% of cervical cancer cases worldwide. Scientists later identify additional strains, 31, 33, 45, 52, and 58, that also pose a significant cancer risk. This revelation sparks an urgent mission: create a vaccine that can stop HPV before it has a chance to damage or transform our body's cells. Fast forward through countless experiments, collaborations, meticulous research, and countless clinical trials. By 2006, scientists and public health experts celebrated a milestone: the first HPV vaccine, designed to help the immune system recognize and neutralize specific strains of the virus known to cause the majority of cervical cancer cases. Initially, the HPV vaccine known as Cervarix targeted HPV types 16 and 18. Then the vaccine known as Gardasil was developed to target the 16, 18, 6, and 11 HPV strains. Soon, an expanded version of the vaccine known as Gardasil 9 arrived, offering protection against 9 HPV types, a significant breakthrough and milestone in HPV vaccine research that offered a much broader protection against several important HPV strains. This broader shield offers significant defense against a wide range of high-risk HPV strains that can affect both men and women, leading to the widespread recommendation that boys and girls alike receive the vaccine. The beauty of this breakthrough lies in its simplicity. Inside each dose of the HPV vaccine are virus-like particles made from a single component of HPV's outer shell, the L1 protein. The L1 protein is the major capsid protein of the human papillomavirus, HPV. It forms the outer shell of the virus and is highly conserved among different HPV types. L1 is responsible for the structural integrity of the virus and facilitates the initial attachment to host cells during infection. Incorporating the L1 protein into the HPV vaccine can therefore instruct the immune system to recognize the L1 protein and prevent the virus from infecting cells and causing cervical cancer. Because these vaccine particles lack the viral genetic material, they cannot cause infection. Instead, they train a key branch of our adaptive immune system to spring into action. Specialized type of white blood cells, known as B cells, learn to recognize these particles, rapidly producing neutralizing antibodies. These antibodies are released into the bloodstream, allowing them to circulate throughout the body. When a real HPV virus appears, these antibodies can latch onto the virus before it enters cells, effectively blocking infection at its earliest stage. Yet there's another layer of immune defense that comes into play if HPV manages to slip past the first line of antibody protection, or if a person already carries the virus. Other specialized white blood cells, known as T cells, particularly cytotoxic CD8 positive T cells, can also target the virus. These specialized cytotoxic T cells seek out and destroy body cells that are infected with the virus, preventing the infection from spreading and reducing the likelihood of progression to cancer. Although the vaccine's primary purpose is prophylactic, it's most effective at stopping new infections through the production of neutralizing antibodies. This robust T cell response can offer an added layer of immune protection. By enhancing immune surveillance, the vaccine may help the body clear lingering HPV-infected cells more efficiently, though it cannot fully reverse advanced cervical lesions once they have developed. Today, scientists meticulously track the vaccine's beneficial impact. There is a notable decline in HPV-related infections and associated precancerous lesions in areas with high vaccination rates. Early data clearly demonstrates a lower incidence of cervical cancer among younger generations who have grown up with ready access to immunization. This demonstrates the success of the HPV vaccine in reducing rates of cervical cancer. Epidemiologists also investigate how vaccination affects HPV types beyond those most prominently linked to cervical cancer, hoping to reduce other HPV-related cancers, like those of the throat, anus, and genitals, in both men and women. The long-term vision is to dramatically reduce, if not eliminate, HPV-related cancers worldwide, a goal that grows more realistic as vaccination rates climb and next-generation vaccines are developed. Yet the success of this vaccine depends on education and awareness. When more people understand how the HPV vaccine works, why it's safe, and how profoundly it can reduce cancer rates, they're more likely to embrace this remarkable scientific, medical, and public health achievement. By combining the power of neutralizing antibodies with the support of T-cell immune surveillance, the HPV vaccine can instruct the immune system to stand on guard against a virus that was once largely overlooked. Standing on the shoulders of decades of research and dedication, we can see a future where HPV-related cancers no longer claim so many lives. It's a testament to the power of immunology, collaboration, and unwavering curiosity in the quest to combat some of humanity's greatest health challenges. Vaccines remain one of the most successful and safe medical interventions and disease prevention strategies. They are a testament to the benefits of scientific and medical research. Thank you for watching. If you enjoyed the video, please like and share. Join us at Science Curious.